Welcome to this Cellar demo of the basic analysis pipeline, which can be applied to all single-cell data. To begin the analysis, we need to first load the dataset. We click the Load the Data button, and then we can either upload our own dataset or select a server dataset. Now for this demo, we select this single-cell RNA-seq dataset as an example. After loading the dataset, the number of samples and features will be displayed. And then we need to do dimensionality reduction. Here we provide multiple methods as well as parameters. The user can either click Run to use the default option, or they can try different methods and parameters and see which one works the best for them. The dimensionality reduction will get the low-dimensional embeddings of the data, which will be used for most subsequent data analysis, such as clustering and differentially expressed gene analysis. And it will also get the 2D embedding, which will be used for visualization. Now the dimensionality reduction has been finished. You can see this 2D scatter plot of the cells. Each point is a cell. And then we need to do clustering for getting the label of these cells. Again, we have multiple methods and settings. We will use Leiden clustering for getting the labels. This is our default option. And Leiden clustering with the default parameters will usually generate more clusters than the ground truth. So we might want to merge some clusters into a single one. To do that, we need to select this group of cells using the lasso tools and first define them as a new subset. To define them as a subset, we go to the Annotations panel and enter the subset name and click Store. And once they are stored, we can select them in this drop-down menu and use the merge function to merge them as a single cluster. Now these cells has been merged. And if for some reason we believe this cluster is good, but we're not sure about the rest, then we can use the constrained clustering algorithm. We go to the clustering and semi-supervised tab. We select a cluster that we want to fix, in this case the subset that we just defined, and then we click Run. This will fix the clusters that we just selected and recluster the rest. Hopefully the clustering of the rest will be improved. And then we show how to do differentially expressed gene analysis. We go to the Analysis panel and select the cluster that we want to find D genes for. In this case, we're going to find the D genes for this cluster that we just defined against the rest of the cells. And after clicking Find D Genes button, after a while there will be a table showing the results. And we can do some visualizations for these genes. For example, if we copy and paste this first D genes here and select a visualization method, for example plot expression, then the color of the main plot will be changed to indicate the expression level. Brighter color means higher expression. In this case, we can see that this D gene is indeed expressed higher in the cluster that we just selected. And we can also see the violin plot for this gene. Also, we can see that it's indeed expressed higher in cluster 1, which is the cluster that we just selected. Another visualization that we can do is the heat plot. For example, if we select multiple genes, and click heat plot, this will generate a heat plot for these 3 genes. Again, we can see they are expressed higher in the cluster that we just selected. And if we click this clear plot button, the color in the main plot will again be used to indicate clusters. Then we can do enrichment analysis. First, select a marker list, for example cell type, and choose the top genes to use and click Run. A table will be used to show the results. According to these results, we might want to do cell type annotation. For example, if we want to annotate these clusters as kidney B cell, we can copy it and go to the Annotations panel, select this cluster 1, type the cell type, and click Store. Then the annotation will be stored here. You can also view the annotations here. For more details about all the functionalities, please check out our documentation by clicking the buttons at the bottom of the page. Thank you for watching.